Good morning, students. We will continue the topic chemistry of proteins, competition number 5.2, and today's subtopic is structure function relationship of proteins, in which we are studying taking three examples: myoglobin, normal hemoglobin, and abnormal hemoglobin. In abnormal hemoglobin, we are taking the example of hemoglobin S. Function of proteins are maintained because their ability to recognize and interact with variety of molecules. The three-dimensional structure conformation of protein provides and maintain the functional characteristics. Tertiary and quaternary structure of proteins are three-dimensional structure where functional groups came close to each other, which are situated away from each other in a polypeptide chain. And this is diagrammatically represented like this. So here it is a diagrammatic representation of an enzyme protein, and there you will see so many different amino acids with their functional groups have come close to each other at the active site of the protein, and actually these amino acids are far away from each other. If you see this number, A5 means fifth amino acid, and A100 means 100 amino acid, A300 means 300th amino acid in a polypeptide chain. So all these amino acids have come close to each other at active site with their functional group, and this is possible in tertiary structure of proteins. Therefore, tertiary structure or three-dimensional structure of protein is a functional protein. The structure functional relationship in proteins can be studied by two facts. First is the three-dimensional structure of protein in turns depends on primary structure of proteins. So any difference in primary structure of protein produces changes in three-dimensional structure of proteins. And second, quaternary structure of protein provides functional advantage to the proteins. So our today's lecture is focus on these two things. So we are studying these examples of proteins to demonstrate structure function relation in protein is hemoglobin S, hemoglobin A, and myoglobin. First is the structure function relation of protein can be studied by, first thing is three-dimensional structure which depends on primary structure. So any change in the primary structure produces change in three-dimensional structure of proteins. This fact can be studied by taking example of hemoglobin S or sickle cell hemoglobin. So what is hemoglobin S? In hemoglobin S, a single amino acid substitution in the primary structure of protein at 6th position of beta chain alters the structure of hemoglobin. So this is the alteration occurs in hemoglobin S. Basically, there is mutation in DNA, in the code of DNA, where CTC is mutated to CAC. Therefore, while transcription, the code on mRNA changes from GAG to GUG. Therefore, during translation at 6th position, normally glutamic acid is present in beta globin chain of hemoglobin, which is hydrophilic. Due to change in the code, that glutamic acid is replaced by a hydrophobic valine, and it causes localized stickiness on the surface of the molecule. So this is the diagrammatic representation of hemoglobin S. On the upper side, I have shown you oxyhemoglobin A and deoxyhemoglobin A. So if you compare the difference between oxyhemoglobin A and deoxyhemoglobin A, on deoxygenation, hemoglobin A develops cavities. Now compare the structure of hemoglobin S, oxyhemoglobin S with oxyhemoglobin A. Now this oxyhemoglobin S has already a protrusions on it which are not present on oxyhemoglobin A. This hemoglobin S, when deoxygenated, it forms cavities like deoxyhemoglobin A. Therefore, deoxyhemoglobin S has protrusions as well as cavities. These protrusions are called as sticky patches. Now, when these deoxyhemoglobin monomers come close to each other, then the protrusions of one molecule of deoxyhemoglobin S fits into the cavities of another molecule of deoxyhemoglobin S, and in this way they will polymerize. And when these deoxyhemoglobin S get polymerized, it mechanically disturb the shape of RBC from normal biconcave to sickle shape. So this is the mechanism of sickling. So same is written here. Sickling occurs under deoxygenated state because in deoxygenated state of hemoglobin has protrusion on one side and cavities on another side, so that many molecules of deoxygenated hemoglobin S can adhere and polymerize. Hemoglobin S can bind and transport oxygen normally. When it is a monomer, it can transport oxygen just like hemoglobin A. This single amino acid substitution leads to polymerization of hemoglobin molecule inside the RBC. This causes distortion in the shape of RBC cell into sickle shape, and RBCs look like this. Next is the structure function relationship in protein can be studied by quaternary structure of protein, which has some functional advantages. This can be studied by taking example of hemoglobin A and myoglobin. Hemoglobin A transport oxygen to the tissue. Hemoglobin A is a tetramer protein made up of 4 subunits. Each subunit is made up of heme and globin chain, either alpha or beta. So it possesses quaternary structure. First is cooperativity effect of hemoglobin A. Here, if we see, binding of oxygen molecule to one hemoglobin facilitates the binding of oxygen molecule to another subunit. Diagrammatically, this can be shown like this. Here on the left side I have shown a hemoglobin molecule with 4 subunits. Oxygen has bind with one subunit. As soon as oxygen binds with one subunit of hemoglobin, the remaining 3 subunit shows increased affinity towards the binding of oxygen, and they immediately bind with the available oxygen. This effect is seen in lungs when hemoglobin becomes oxygenated. Next, when one similarly release of oxygen by one heme subunit facilitate the oxygen release by another subunit. This can be diagrammatically shown here. On the upper side I have shown a oxygenated hemoglobin A molecule with 4 oxygen molecules. As soon as one oxygen molecule is released from hemoglobin, then another 3 subunits also loses the affinity towards oxygen, or they show decreased affinity towards oxygen, and they immediately release the oxygen, and this effect is seen in tissues. So both this cooperativity effect helps in binding the easy binding of oxygen with hemoglobin in the lungs and easy release of oxygen to the tissues. Next functional advantage of quaternary structure of hemoglobin A is Bohr effect. Binding of H plus ion and carbon dioxide promotes release of oxygen from hemoglobin A. So this allosteric interaction is physiologically important and is termed as Bohr effect. Now here H plus ion and carbon dioxide concentration is more in tissues. So as soon as hemoglobin binds with this, it immediately releases the oxygen to the tissue, and this is possible only in quaternary structure of protein. 2,3-bisphosphoglycerate effect. This 2,3-bisphosphoglycerate is synthesized in RBCs. It is the product which is synthesized in glycolysis. Now, when this 2,3-bisphosphoglycerate binds to the beta chain of hemoglobin A, it promotes release of oxygen from hemoglobin A. This is also very important effect to deliver the oxygen to the tissues. In case of anemia, high altitude and other ischemic cases, we see that the level of 2,3-BPG in the body increases just to release more oxygen to the tissues. So this allosteric interaction is physiologically important and it is seen in quaternary structure of proteins only. It is not a Bohr effect. Next example is myoglobin. Myoglobin is present in muscles. I have given the concentration of myoglobin in various muscles. In skeletal muscles, 2.5 grams of myoglobin is found in 100 gram of skeletal muscle. In cardiac muscle, 1.4 grams, and in smooth muscle, 0.3 grams of myoglobin is present. The function of myoglobin is to store oxygen. Myoglobin is made up of one heme and one globin chain, so it possesses tertiary structure. One molecule of myoglobin can combine with one molecule of oxygen. Myoglobin stores oxygen which can be released during ischemia, that is during exercise. Function of myoglobin is different from hemoglobin A. As we have seen, hemoglobin acts as a transporter of oxygen, whereas myoglobin, it is also having one heme and one globin chain, but it does not have quaternary structure and therefore its function is different. And myoglobin shows high affinity for oxygen than hemoglobin. Now this high affinity of oxygen of myoglobin can be demonstrated by a fact which is given here. At PO2 30 mm of Hg, which is seen in tissues, myoglobin is 90% saturated. Means only 10% of myoglobin have released oxygen to the tissues at PO2 30 mm of Hg. At the same PO2 30 mm of Hg, hemoglobin A is 50% saturation. Means 50% of hemoglobin have released their oxygen to the tissues. So at 30 mm of Hg, PO2, myoglobin releases only 10% of oxygen to the tissue and hemoglobin releases 50% of oxygen to the tissue. So this shows high affinity of myoglobin towards oxygen. But during physical exercise, when PO2 in the muscle lowers to 5 mm of Hg, then myoglobin releases all oxygen to the tissues. Higher affinity of myoglobin for oxygen make it suitable for storing oxygen. This higher affinity of myoglobin for oxygen is due to absence of cooperativity effect, Bohr effect and 2,3-bisphosphoglycerate effect. These effects are not seen in myoglobin because it does not possess quaternary structure. So this is the structure function relationship between function relationship of proteins. Thank you very much.